everyone here. Professor Dame Carol Robinson is the previous president of the Royal Society of Chemistry and Dr. Lee's Professor of Chemistry at the University of Oxford. Professor Robinson has a very interesting career pathway into research. She completed her graduate education while working full-time in industry. She was subsequently admitted to the University of Cambridge, where she completed her PhD in just 2 years. Following an 8-year career break to begin raising her 3 children, she returned back to research at Oxford, and in 2001, she became the first female professor in chemistry at Cambridge, subsequently returning to Oxford in 2009 to take up the chair of Dr. Lee's Professor of Chemistry. Her research on mass spectrometry for determining the 3-dimensional structures of proteins has attracted numerous international awards and prizes, including the Royal Medal A from the Royal Society, the Stein and Moore Award from the Protein Society, and the Field and Franklin Award from the American Chemical Society. For her contributions to science and industry, Professor Robinson was awarded the Dame Commander of the Order of the British Empire (DBE) in the 2013 New Year's Honours. Let us welcome Professor Dame Carol Robinson to deliver her lecture, Mass Spectrometry from Ribosomes to Receptors. Professor Robinson, please.
Thank you very much for your excellent introduction. I regret that I can't see anybody in the room, but I will be much better in real life, but unfortunately we are in this situation, so I'm going to try and make the best of it and carry on. And I thought because you're all sort of starting out in your scientific careers, it would be good to sort of trace the journey rather than just talk about the results, because I chose a path that wasn't necessarily very well accepted early in my career, and I think that has a lot of challenges and also a lot of rewards. I'm going to try and bring out the highs and the lows of a research career where you choose a different path. So I've called it From Ribosomes to Receptors because that's kind of where I've been. I've been starting with very small proteins and building up to much larger assemblies, and then trying to move into membrane proteins because these are some of the most challenging molecules that we can study in structural biology. And I thought I would start with my, sorry, this is horribly wrong again, with my long-term mentor and good friend, Christopher Dobson, who some of you may know because he was in the chemistry department at Cambridge, and when I was a postdoctoral student, he was the person who always encouraged me to sort of follow my dreams and to reach for things that might be considered a bit outside of what was possible. So I'm extremely grateful to Chris for all of his help and support, and very sadly he passed away a year and a half ago now. So, but without him, I don't know where my career would have been. So everybody should get a really great mentor who will help encourage them and support them during their research career. And in Oxford, when I was coming back from my maternity leave, I came into Chris's lab, and we found a way of looking at protein folding using mass spectrometry. Now, I'm sure you know about mass spectrometry, but in a way whereby you can characterize a peptide or a small molecule, you measure its mass very precisely, and you can break it a bit and tell something about its composition and its structure. We wanted to do something quite different. We wanted to take a protein and keep it in a folded state, so not denature it, not break it, not destroy it, but actually preserve it very gently. And when we did this, we found that we could observe protein folding intermediates on a folding pathway. And in this particular case, this is the protein hen egg white lysosome, which of course is in your morning eggs. We followed its folding and found that it had intermediates on that pathway that couldn't really be seen in other techniques. So this was for us a sort of breakthrough moment, that we could track a folding intermediate which was sort of halfway between the folded and the unfolded states. But of course, a lot of people didn't really believe that, because you're in the gas phase, and everything you know about a protein and everything you learn about a protein is that it requires solution or cellular environments to stay folded. So this was in the vacuum of a mass spectrometer and was considered a bit unusual for quite some time. But for me, what was important was not just about, not just the folding, but also the binding. So the next set of experiments that we did was to study folding in the presence of a ligand. And in this case, we saw that the small protein, acyl-CoA binding protein, folded much faster when it was in the presence of its ligand. And one day, sitting in front of the mass spectrometer, I noticed that the folding coincided with the binding of the small ligand. So if you imagine you're sitting there spraying away in your mass spectrometer this protein, and you monitor how it folds, but at the same time you see that it binds to its ligand, and then that increases the mass by the composition of the ligand. So you've seen both a folding and a binding event. And to me, that started to make me think that the structure of a protein in the gas phase and its interactions could actually be preserved. And so with my first ever research grant, I proposed to build a mass spectrometer that it would allow me to go to very high molecular weights so that I could see if these folding proteins actually survived within assemblies. So it was a very sort of unusual thing to request, because at that time all the mass spectrometers were being geared for proteomics, which of course is an extremely powerful technique. And it wasn't considered that this would be a particularly productive line of research, and perhaps it wasn't really worth spending a lot of money modifying a machine. But I was so convinced that this was what I wanted to do that I put all my effort into persuading somebody to help me build and to purchase such an instrument. And I'm not sure, because I can't see all the participants, but I'm not sure if this would be of interest to you. But this was the original design. So we have a very small capillary here from which we electrospray our protein mist into this region here. We have an ion tunnel, a high master charge quadrupole, a Clausian cell, and then a time of flight, which is in here, and it's a sort of V-shaped thing, so that the protein complexes fly down and fly back up again. And then they get recorded on this detector here. There's lots of high voltages and lots of excitement. Used to be very exciting when I started in mass spectrometry, because there was often flashes and bangs and things would not quite work. So I think that was why I became captivated with the whole thing. It was always so exciting to see if you could get a spectrum, because it was never a given. And one of the complexes I first started to look at was transthyretin. Now, transthyretin, you may have heard of it. It's something that transports retinol and thyroxine around the body, and it's present in plasma. And I wanted to not just do proteins such as hen egg white lysozyme, which are extremely abundant and very well behaved and well characterized, but to try to map other interactions which are much less well studied. And for this, I isolated from chicken plasma a protein which binds to vitamins and hormones simultaneously. And then what I did was to sort of simulate all of the different compositions that you might anticipate from the plasma of a chicken and sum it all up. And this is showing in a very, very simple way the composition. This is the raw data, sorry, and these are all of the different components. So this is the spectrum that we would anticipate from this central region, binding to one protein, binding to another, and binding to retinol. And I wasn't very confident in those days. So I remember at the end, I just put, well, maybe this will be a general method for analysis. And it's truly now widespread around the world. So I'm delighted to be telling you about this method, and it's not just general method that occasionally we use. It's actually very widespread now and a recognized field in its own right. I also published this in a journal which I think subsequently closed down and is not very well known. So I think I was just suffering from being constantly criticized at that stage and not being very confident in what I was doing. I've changed that now, I'm pleased to say. And the next spectrum represented a turning point for me. I was very fortunate to be working with Venki Ramakrishnan at the LMB, and he, as you know, got the Nobel Prize for his ribosome structures. And he had obviously some preparations that we were able to fly. And we say fly because we're just putting them into the gas phase and they're flying through the mass spectrometer. And we found that we were able to discover the composition of this stalk region. And this had resisted crystallography because, as you can see, it's extremely dynamic, sort of folding. And this region was never going to be on the crystal structure. So this bit was crystallized separately and then docked back in. But we could see its composition because as the flying ribosome went through the mass spectrometer, this 96 kilodalton stalk was released. And this is the stalk region here. And it was much larger than anticipated because it was thought that there were only 4 of these red proteins. And in fact, there were 6. So mass spectrometry, although cannot give you such a beautiful crystallographic structure, it can really add stoichiometry, which, as you know, is critical to a lot of these, the function of a lot of these complexes. So if we think about how these complexes go through the mass spectrometer, is it the case that they actually look anything like we think they do as they fly through the mass spectrometer? And how are we going to determine this? This is a movie that was made for me by one of my postdocs. Coming through the mass spectrometer, we added in this additional segment here, which we call an ion mobility separator. And what it does is, as the protein comes towards you, you see it sort of buffeting with these small molecules. It's having these collisions which separate according to the collision cross-section. And that's in this region here, what we call the drift cell, which is like a sort of cloud chamber, I suppose you could say, because if something is very tightly folded, it can go through there much more quickly than some sort of lumbering, much larger complex. So if you think of ion mobility, it's giving us a new dimension, because my fist here has a mass to charge ratio that would be measured. But if I open it and have an open hand, you can see that that's going to take a lot longer to go through the mass spectrometer. So we have a way now of distinguishing open and closed conformations of complexes. And actually in the lab at Cambridge, 2 of my postdocs, Maggie McCammon and Brandon Ruttelow, discovered that we could separate a complex that was a collapsed ball or a ring-shaped donut. And they were in the basement of the chemistry department in Lensfield Road, and they managed to separate these 2 shapes so they have the same mass to charge but a different collision cross-section. They worked so hard together that they subsequently got married and now have 3 children. So science does all sorts of strange things and also changes your life in lots of ways. So this is their work. And fast forward another 5 to 10 years, another milestone for me was to try to look at membrane protein complexes. As I've said, they're extremely challenging for structural biology. And if you think about what's challenging about them, it's because we've got to try to keep them in the membrane because they are so difficult to study outside of the membrane, and you have to have membrane mimetics. We realized that this was also the problem because in the mass spectrometer, you first have to be in solution, but then if you could get them into the gas phase, then you should be fine because all the solubility issues that you normally suffer with would now not be required. So, and this is the work of John Hopper, looking very smart there, and he showed that if we covered it in detergent, this is an ABC transporter, B2UCD, if we covered it in detergent and introduced it into the gas phase, it would look ugly because, I call this ugly, because all of the detergent would be stuck and you wouldn't be able to resolve the peaks because there wouldn't be one species, there would be a lot of species coated in different numbers of detergent molecules. But what he found was that if you can sort of tune it to the sweet spot of the mass spectrometer, out of that mess comes these beautiful peaks. I'll stop it there. And here now we see the intact oligomer, a little bit of fragmentation, both high and low. And if we continue, you can see that we go too high and the complex starts to break up, and then back lower again, and you'll see that it becomes less well resolved and you just get that horrible hump again from which you can tell nothing because you have too much detergent. So what we imagine that we're doing is actually shaking off all the detergent and releasing this membrane protein complex in the gas phase of the mass spectrometer. So what would you like to do if you could do this? Well, I've always been fascinated by the ATP synthase, and I know that in your session of talks, you've had John Walker give a talk about energy and life, which is all about ATPases. I mean, it might be before you were in the society, but I saw it on your website. And he, of course, won the Nobel Prize in Chemistry for his work on the ATP synthase. And this is an enzyme that sits and rotates in the membrane and synthesizes ATP or acidifies cellular compartments. So incredibly important. And for me, very exciting. I was working with Daniela Stock and we could eject this whole complex, which to me, I never thought I would see the day that 29 subunits could come out of a membrane environment and fly through the mass spectrometer and stay intact. And that's what this particular series here, these peaks here, at 12,000 mass to charge, 57 charges represent the mass of this ATP synthase. One of the very interesting and perhaps not so surprising aspects of this research was that it came with its lipids associated with the ring. And although that's not so surprising because look where it lives, we weren't really expecting it. So we almost made a very bad mistake. But we found that if we added in the correct number of lipids in this particular ring, the mass fitted perfectly. And you can imagine this is quite a difficult spectrum to interpret because you don't know how many lipids you have. You don't know if you have any nucleotides binding here. And we didn't actually know the subunit stoichiometry. And I printed this. It's a kind of happy Cambridge memory for me. I printed this and put it on my desk. It was I made it about a meter long. And every day I tried to sort of assign a bit more because it was a very challenging spectrum. We had it at all different energies, and different energies give you different picture. And it's a very time-consuming activity, but also very rewarding. Of course, you can do computer algorithms to sort these things out now, but there's nothing quite like doing it by hand, because when I started, I didn't really understand the mechanism of how it was going to break in the gas phase, and so it wasn't really possible to program that until you have enough experience with these molecules to understand their fragmentation pathways. So still, although my students think it's a bit of an odd thing to do, I still like to try and interpret things by hand after they've done their computational assignment, and I can sometimes pick up mistakes, so there's always room for going back to basics, I say. Okay, but that really started us thinking about why some proteins require lipids to be stable in the gas phase and some don't. And this is work of Kalur Gupta from my group. He's now associate professor at Yale, and he found that if you go into the PDB and look at all the alpha helical membrane proteins that have been crystallized, you could plot the number of salt bridges, their buried surface area, and have this kind of colored thing whereby black means that you have no salt bridges, and this is your buried surface area on this axis. So anything that doesn't have many salt bridges or much of a buried surface area, so if you think of surface area of interface, then something like this without any lipids gluing it might be a bit more fragile than something like this with lots of lipids in here. So we had this hypothesis that if it was very fragile like this, maybe the lipids would be required to glue these proteins together. And so we chose two in this corner, the sodium proton antiporter in two different species, and we expressed them actually in the same lipid environment, and we saw something very interesting, very different behavior. So on the left-hand side, this is in E. coli, and on the right-hand side, this is in a thermophilic species, so much more stable. So this is the NAPA, which I showed you had salt bridges and overlap. This one, no salt bridges, no overlap. And you can see, I hope you can see, you may not like my spectra, but of course I think they're amazing. But if you look here, there are just the peaks of the protein with 10 protons, 11, 12, 13, 14. If we look here on this side, you see a mess completely because you have all of these protein peaks, and they have different numbers of lipids holding them together. So this is a very complicated spectrum. This is a very simple one. And this is really showing you that these lipids are required to glue these interfaces together in this detergent material. And so with this, we plotted the delta G values of breaking these interfaces, and we could see that this one, which wasn't so stable, had lipids that bind in here, and this one is much more stable, doesn't require lipids. And so we thought, well, we have now a predictive scale for lipid dependence. And if we look at this, it's actually very similar to what we see in GPCRs, which often crystallize as dimers, and they have this sort of inviting gap here, which we thought might be populated by lipids, as we've seen here. And as you can see here, there's a tiny green bit, which was the start of some density for a fatty acid. So we hypothesized again that perhaps these G protein coupled receptors, which I hope you might have heard of, they're very important in all sorts of cellular signaling processes, major drug target in the pharmaceutical industry because of their roles in touch, pain, sensation, and lots of different diseases. So we reasoned that these might also be lipid-mediated dimers, according to our hypothesis. And these the two heroes here, Jason Yen and Idi Liko, and they isolated this adenosine receptor, and it was stabilized with a nanobody. And you can see we have GS, G alpha, G beta, and G gamma. So we have this pentamer. And Jason here, he worked for a number of evenings and nights to get this spectrum. And I do remember not being wildly excited by it because it doesn't actually look all that beautiful. But it was an amazing feat because a GPCR flying with its signaling cascade intact had not been achieved before. And we always thought it would be so tricky to stabilize in the gas phase because a GPCR sort of floats around and picks up signals, and it's very very unstable sort of dynamic complex, which is hard to kind of preserve in the gas phase. So when he got this, of course we weren't super excited, but we realized that this spectrum is not great. If we isolated one charge state, so using a sort of tandem mass spectrometry experiment, you can just pick off one particular ion and then break it and say, what is the composition of this? Which is what he's done here. He's isolated this ion and broken it in the mass spec, and you see then it yields this beautiful spectrum of the receptor and all of the nanobody and signaling components here. And as I mentioned, we were interested to see if lipids were important in stabilizing a dimer. We didn't actually find a dimer, but we did see lipids being important on this receptor. And we postulated that they might be important for binding to these downstream signaling components. So we set out to observe this. And then a very nice thing about mass spectrometry is you can ask questions and postulate ideas quite quickly and then go and check to see if they're right. So we reasoned that it would be this PIP2. We added different PIPs and we mixed them all up together. And so you can do these kind of competition experiments where you expose the receptor to the different types of PIP lipids. We then simulate. I'm sure you know all about simulations. You put your receptor in this lipid box and then you expose it to all these different forces and monitor where PIP2, in this case, where it ends up binding on the receptor. And we were happy to find that it bound on the inside of the receptor, which was important for signaling because this is where the receptor signals to its G proteins and passes on the chain. These are the PIP2s that we observed. And this is how it works. You have an agonist that binds here. It engages with these and you get this GTP turnover, GDP release. And this was the complex that we were able to trap. But we found, using a mini G complex and an agonist, that we could observe these PIP2 lipids binding and stabilizing this complex. So we discovered a signaling molecule, a lipid, that was changing the ability of the GPCR to signal to the downstream G proteins. And this, for me, was a very exciting moment. I remember it very well because we could see actually TIP2 of these lipids was the optimum number for coupling. And we realized then that we had this amazing platform. I say amazing, maybe I'm biased, amazing platform for studying GPCR pharmacology. We could look at a ligand binding, we could look at different polymorphisms of the GPCR, could look at how the lipid environment affected things, we could look at allosteric modulators, PTTM, modifications, post-translational modifications such as phosphorylation, and we could look at all of the different flavors of the G proteins. These are some agonists that you may have heard of: isoprenaline, which of course binds and then increases coupling, norepinephrine, and a partial agonist, dobutamine. So here we're seeing a little bit of coupling, but a lot of the free receptor. So we had a sort of pharmacological readout of what was going on here. And we could also look at orphan receptors, which is a very exciting opportunity, because you could look to see what ligands or metabolites might be trapped within these receptors, and that might give a clue to the function of some of these orphan receptors. And because this was a sort of exciting development in the field of GPCRs, it attracted investment, which was exciting for me. I'd never had a company before. And so a company was born called OMAS Therapeutics, and the remit here is to discover modulators of G protein coupling via our sort of unique platform, which tells us about the role of lipids, modifications, drug binding, different coupling, and we could do screening of, we now have about 170,000 molecules, So clearly we can't do this in the lab. It's not appropriate for PhD students. So this has now been spun out onto the Oxford Science Park and has about 40 employees. So I'm very happy to talk about that if you're interested. So we discovered that PIP2 bridges are formed between this G protein and the GPCR, and they're shown here in green. And quite interestingly for me, Bran Kabilka, who you maybe know, had the Nobel Prize for Chemistry for his X-ray structures of GPCRs. And he told me that his group did a group meeting on our Nature paper, and they weren't convinced, he said, because mass spectrometry is all about charge. Maybe it's just a sort of charge, charge sort of complementation happening in the gas phase of the mass spectrometer. But then subsequently, I was delighted to see in his structure in 2020 that PIP2 featured in his binding of the neurotensin receptor to arrestin here in pink. And then just recently we collaborated with a group in China, Eric Zhu's group, and we found again this PIP2 lipid, and that's coming out very soon, just accepted. So although we discovered it using mass spectrometry and people were not always convinced by mass spectrometry, subsequently it's turned up in two structures from two different groups in two different GPCRs, so I'm excited by that. But we also noticed that lipid binding, stoichiometry, and all these things about membrane proteins are very dependent on how they're treated once you've got them out of the membrane. You can see here, this is a massive nanodisc or detergent micelle. Can't quite remember which way they did this one. But obviously, if you change this, it may be that you wouldn't see the PIP2, or you may see a different stoichiometry. And we started to think more deeply about how detergent affects the subunit stoichiometry and the lipid binding properties. And you only have to look through the literature to notice that there are a number of examples where stoichiometry changes. For example, this is the patched receptor, published in Science as a monomer, later in Cell as a dimer, and then in Nature Communications as a tetramer. And there are other examples, I won't go through them all, where it seems that there's some differences in stoichiometry which tend to be somewhat dependent on the detergent environment in which they're sitting, or in the membrane mimetic. And that really made us think that what we would like to do is to actually not take them out of the membrane. And this is a slightly crazy idea, because we've got to think about how we treat the membranes and then how we extract the protein complexes from them. So we have here a vesicle preparation, which we can do either via excretion, cell press, or osmotic shock. And we can get these interesting shapes. This is the work of Trorev. And then we need fairly high extreme conditions in our Orbitrap UHMR, and we break off some of these pieces, and then we look at the charged complexes. And Lindsay Baker helped with preparation of the membranes, and I'm showing you here an E. coli BAM complex sitting here, and it comes out complete with its lipids, which we see. You can always tell the lipid peaks because they're always quite diffuse, because lipids are not just one lipid, they're a family of lipids. And so we saw the BAM complex with these lipids, and we saw OMP-A, we saw all sorts of other things. I won't go through all of them, but just to say that we were able to observe these lipids binding very nicely, and you can see that these are the native lipids that came with the complex as it was ejected from E. coli. And we've recently seen these again in a paper just about to come out in Nature, just accepted, whereby darabactin, an antibiotic discovered in soil, can target this complex, and its binding is augmented by this cardiolipin binding here. So very excited to see that. These are the cardiolipins. This is my colleague Saima Khalid, and you see here 2 and 3 cardiolipins, and these support the binding of the antibiotic that targets the BAM complex. We then looked at, again, one of my favorite complexes, the rotary ATPase, which I talked to you about earlier. And here we saw with its SEC-YEG translocon actually caught in the act of insertion into the membrane. So I think what's exciting about this method is that you often capture things going on within the membrane. I'm not going to talk about all the spectra, but just to say, remarkably, we were able to capture some of these multi-drug efflux pumps which span both membranes. So clearly it's giving us fragments of membranes, and we're not quite clear which bits are going to be preserved and which not. It's very exploratory, but the fact that these bridging molecules survive, to me, was exciting. In the membrane, in the mitochondrial membrane, spectra are very complicated, but we were able to see a nucleotide translocator with palmitic acid caught in the middle, and various other intact mitochondrial complexes. This one I think looks particularly nice because in detergent, you can see, if you're an aficionado like me, I like nice sharp peaks. These are quite broad, and these are very, very difficult to assign much more beautiful resolution when directly ejected. Not the full complex, we don't see this region here, but we do see all of this, and we can see up to 4 cardiolipins quite well resolved, and we believe that these are integral to this structure. We came up with some artwork because that's what you do, and this is actually the rotary ATPase coming out of the membrane. So actually, I think it's going in not quite the way I would imagine, because I think the soluble head should go in first. But it's a piece of artwork, so we're happy with that. And this is the high electric current that's used to extract. So we hoped it would go on the front cover, but they chose this avocado sandwich. A bit disappointing, but we use this all the time, so no worries. But it made us think about other solute carriers that are involved in the transport of small molecules across membranes, because there's lots of solute carriers, and it's very hard to see what the small molecule cargo of these complexes might be. And so with Katerina Dewar and the Resolute Consortium, we've been looking at some of these, and this is SLC 25A13. They don't have particularly friendly names. And extracted into detergent, we see monomers and dimers with cardiolipin binding, various numbers, because of course it's the inner mitochondria and has lots of cardiolipin present. And however, if we use our method of ejecting directly from the membrane, we see this beautiful dimer and small molecule binding. And this is a glutamate transporter, so we believe that these are glutamates caught in the act of transportation. So, and here again we see 2 cardiolipins binding. 2 seems to be the magic number for stabilizing this solute carrier. So exciting, I think very exciting. Currently trying to use site-directed mutagenesis to probe these binding sites, and to look at how it's also seen in databases. So, but how do we for sure identify the small molecule that's within these complexes, because it's very tricky? And what we would like to do is to take these molecules in through our mass spectrometer, and then optionally take them out again, cycle them around and break them up for another round of mass spectrometry, and then bring them back in again and detect them. And this is what we're calling nativomics, for want of a better name maybe. So we have our membrane protein sat in the membrane, we have some small molecule binding, we observe this complex, we release the small molecule, we break the small molecule, and we get a thorough chemical structure. So it's going from a very native-like environment to a broken complex or protein. We can do both and try to assign without any ambiguity what's in the membrane and what it's binding to. And I'm just going to show you one example of this method that we're using in earnest now. And this is a mitochondrial transporter, TSPO, and it was crystallized. And I hope you can see there's a little bit of ambiguity here because the lipid that they fitted didn't really fit too well the electron density. And when we looked at this with our nativomics approach, we saw the spectrum for the intact dimer and the lipid associated with it. And then when we broke the lipid associated with it, we found that it was this particular lipid, and we modeled this into the EM density. And of course, we like to say it fits much better, and you can see that all the head group comes right to the end of the density, and there's some interesting interactions that stabilize this particular lipid in that pocket. And now we have not only saying what the lipid is, but actually the unsaturation and the different tail lengths. So you can actually get the kinking of the chains, which is very exciting to me. And I grew up on these small molecule mass spectra, so I like to think that I can impress my students by interpreting these spectra again. So I'm going to show you now a timeline for mass spectrometry, like my timelines. So when I started, I was looking at folding intermediates, and we were keen to go into membranes, but we could only really do those membrane proteins that bound on the periphery of cell membranes. This was one of my favorite molecules for a long time, the intact GROEL14mer, 800 kilodaltons molecular weight. And when we first got it to fly, we were excited, but now it's become sort of a tuning compound to make sure the mass spec is working okay. These are contributing to different designs of mass spectrometer to preserve intact protein complexes. For me, the first spectrum of a membrane protein complex, which has both soluble and transmembrane parts. I mean, you could sometimes see aggregates in detergent, but actually seeing a real complex, I think, made me really confident that this was going to work. I mentioned about the different shapes of complexes that we'd managed to see. The Atp Synthase is one of my all-time favorites, how lipids affect membrane protein structures, and then GPCRs in 2016. And although these look very simple, they were incredibly difficult to preserve because of their very floppy, labile nature. And then, as I mentioned, mitochondria. But I'm going to finish by showing you sort of a full circle story, because I started by telling you about the ATP synthase in bacteria. And this is now isolated from a rat, just a single rat brain, not by me, but by others. And they really wanted to see if we could get a synaptic ATP synthase from brain, and we can distinguish that from other ATP synthases by the composition. So this is where it sits. It's involved in neurotransmission. And this, to me, again, quite remarkable because it's isolated from a rat brain, and you may think this is very ugly, not at all interesting. But for me, it was fascinating because each of these peaks can be assigned to a composition of something. And if you sort of home in on this region of the spectrum, it's rich with information. It's one of these things, again, where you sit and look at it for a long time and you pick out series and you work out what the composition could be. What we needed to show was that G1 or G2 was incorporated into this complex. It's a very large complex, and the mass differences between these two subunits are actually very small. So to distinguish them, they're just over 40 Daltons, very small number, within the context of a very large thing that's been isolated from a rat brain, to me is still quite remarkable. So we could see at the lower end, actually, that G2 was the subunit that was present. You can see with the red circles, we managed to find this, and therefore to show that actually we did have the rat brain synapse ATPase, and this is its spectrum. And in general, I'm kind of used to the fact that we do all this work and we spend a long time trying to assign every little peak in here. And there's the editor of the journal will say, oh yeah, it's a bit technical. We'll put that in the supplementary material. But this time the editor said, no, that's figure 1. That's such a beautiful spectrum from a single rat brain that it has to be figure 1 in the science paper. So I was delighted by that outcome, because here we have the intact complex, the different isoforms of, different copy numbers actually of the C subunit that exist here. And this was actually disputed originally, but more recently we have come to an agreement that there are different numbers of this C subunit. And here you can see how we've got a lot of V1 subunits coming away. Actually, most people, of course, prefer to see the beautiful structure, but this comes with the mass spectrometry to support it, because it was actually incredibly hard to understand the stoichiometry and the modifications that are present within this rat brain tissue. So with that, I'm going to thank everybody in my group. And this, sadly, I haven't been able to take them out this year, because we haven't had the opportunity to all get together on an away day. This is obviously the Radcliffe Camera in Oxford, and this we have an annual away day where we discuss future directions and give presentations. So this is my current group, my collaborators, and my funding sources here. And with that, I think I will stop, because you've all been sitting patiently for nearly an hour, and ask for any questions.
Well, thank you very much. Thank you very much, Professor Robinson, for this really exciting talk. I see lots of recent research about determining structures and more complex structures using mass spectrometry. That's really interesting. So now we'll open the Q&A to the floor. If you have a question, please kindly unmute yourself. And if you would like to turn on camera, please do also. And over to you. If anyone has any questions.
Sorry, I was just going to try and turn off the sharing option.
Maybe I'll just kick off the Q&A with a first question then.
Sure.
Yeah, actually, if you'd like to stop sharing screen, that's fine too.
Yeah,
I think.
Yeah. Stop presenting. Yeah.
Right. Brilliant.
Thank you.
Right. So maybe I'll just start off. Perhaps maybe you could just briefly summarize how would you compare mass spectrometry to other structural biology tools, such as the old X-ray crystallography, as well as the up-and-coming cryo-EM?
Yeah. Well, nobody can really compete with the amazing results of cryo-EM. I'm very jealous of everything that they have. But I do think that there are complementary roles, and actually bringing together the mass spectrometry with the cryo-EM is very informative in terms of the small molecules that you can't actually see and the much larger molecules where sometimes there's some ambiguity into the composition. So I think the two together is extremely powerful. Do you use cryo-EM yourself?
No, I'm not really doing chemistry, so but
Okay.
I've heard of that technique, and I've read a few
Yes.
papers about using cryo-EM for various structures. So they do give really beautiful structures.
Yeah,
Quite
they're
detailed.
amazing. Yeah, amazing structures.
Hi, Carol.
Hi.
Very nice presentation, first of all.
Sorry, I'm not sure if I'm getting your name correct. Yeah, GN, is it?
So, yeah, it is, yeah. So I was just wondering how this MS technique compares to other mass spectrometry techniques that you can use in proteomics. So I guess, for example, something that I thought about was like if you can see the entire complexes, where does that leave, for example, cross-linking mass spectrometry?
That's a very good question, John. So I think again, it's very complementary with cross-linking. So with cross-linking, you can get residue to residue information. So if you also know the stoichiometry, you can start to build a map. If you just have residue to residue and you're not quite sure how many copies of things that you have, you can see that that's going to be very difficult to model. So I think, like all these things, you need to have all of these techniques going for you to really get an accurate model. So do you use cross-linking?
I'm starting to get a bit involved in cross-linking, but mostly from the chemistry side of it in terms of designing and synthesizing, but yeah.
Okay, that's interesting. Yeah. Well, we also use cross-linking to support some of our models as well, so yeah.
Right, I suppose it's just, is it, I guess with the data that you get, it's very difficult to deconvolute, whereas I suppose cross-linking MS would probably be a bit easier, I suppose.
Yeah, it's more straightforward to interpret, I would say, yes.
Right, okay.
Very powerful. Yeah. We're always needing more cross-linkers, so if you think some more, that would also be useful.
Right, okay, thank you.
Hi, thanks for such a great talk.
Hello.
Hi. So my question is about high throughput mass spectrometry, which I suppose is a little bit different to most of the stuff that you've presented, and maybe a long way off for the sorts of molecules that you're analyzing. But my question is, do you think that it will ever be possible to use so high throughput mass spectrometry of things like GPCRs to do a kind of high throughput binding assay of small molecules?
Great question. That's exactly what we do in the company that I was talking about. So for OMAS Therapeutics, they do that screening. So they have the protein, they have the coupling, the G protein coupling, and then they add the agonist, and then you can screen through and just read out from the mass spec, is it having an effect or not? So we don't only do that, we do cell-based assays as well, but we have this high throughput screening first by the mass spectrometry, and then following it up with other assays. So yeah, it is possible to high throughput. I wouldn't say you would do unknown complexes in that way because there's just too many variables. But once you have your system set up, so if you have a really well-behaved protein, and then you can just add lots and lots of ligands and check for binding, and that can be high throughputed with the robots. And in the company, they have all these robot pipettes and hundreds and hundreds of wells and churn through. And we have ways of marrying that up to the mass spec now in very native-like environments so that they're not unfolding. But yeah.
Impressive.
Yeah, it is. Yeah, it's not the sort of thing for students, so I think it's not really fair because, go screen 170,000 compounds, and they wouldn't. So that's why it has to be in a company.
How would you be able to give a quick flavor of how long it takes to, and how much effort, how many resources it takes to set up one of those assays? Because it would obviously be amazing if you could take any GPCR and just immediately small molecules at it that have very similar structures and detect the binding. But obviously there's a trade-off, because if it takes 5 years to set the experiment up, then it's not so high throughput.
No, it doesn't take 5 years. We would be out of business, I think, if we took that long, because we wouldn't be competitive at all to other methods. So no, we can certainly crank through. The long part is, as you say, it's getting set up. Once we're set up and it's all spraying and it's all happy, it's quite easy. We reckon we can do several thousand compounds in a day, but we would multiplex them in a well. So we have several in one well, and then we get a hit. Then we go back and sort of separate out you look for the masses of the hits, and then you go away and do them as singletons, perhaps, and check the. So you can rapidly screen quite a few things, but then you've got to go in and be a bit more careful.
Yeah, sure.
That makes sense? Yeah.
Yeah, it does. Thanks very much.
That's okay.
I think there's a question
Yeah, somebody
on the chat. Yeah.
Yeah, I recently listened to your Life Scientific, and you mentioned that you were just a postdoc when you came back. Yep, this is all true. While your peers were lecturers. In what ways is the experience of being a postdoc different to that of being a lecturer? Well, I think the way I'd answer that is, while I was a postdoc, I was always receiving instruction from a lecturer or a researcher. When you become a lecturer or a professor or assistant professor, you then dictate your own research. And so for me, that was the big difference. My friends who were lecturers were already leading research programs, whereas I was performing the research that others were leading. So that's the difference between those two things. I mean, I don't regret the time I had out, so it's not a complaint, it's just a difference. And I wasn't either resentful or particularly jealous of that. I was just quite happy that I'd managed to have some time with my children, and quite happy that I did that. Sometimes people say it would be difficult to do now, and maybe that's the case, but I think that's a bit of a shame, because everybody should have the choice if they want to or if they don't want to take time out.
Hello.
Hi, Oleg.
Hi.
Oded. Okay, sorry, I didn't read it very clearly.
That's all right. Yeah, first of all, I want to thank you for a wonderful talk, and to welcome you. This is the Center for Misfolding Diseases that was founded by Chris Dobson.
Nice. Oh, I wish I could come. That looks really...
Yeah, welcome you virtually to the center.
Yeah.
I wanted to ask about the level of purity you need the protein complex to be at, because when we do whole protein mass spec, we use LC-MS to purify the compound, and still we see impurities, and we need some work to figure out what's the impurity and what's the actual protein we're looking at. How do you handle when you have such huge complexes and you cannot denature them? What do you do for purity?
Yeah, I mean, that's a great question, and it's always about the purity of the complex. Everything is, and as you know, if you do any structural technique, how your sample is prepared and handled is the key to everything. So I would, yeah, how do we do it? We have a lot of different ways of cleaning through different centricon devices, different washing methods, different affinity preparations. It can never be too clean is always the mantra, sadly. And just, I suppose mass spectrometry is quite interesting because it's very tolerant of some things, but not at all, so it has different tolerances. And for EM, for example, it might be difficult to have a particular molecule, whereas for mass spec, it can be a different one. So it's a question of getting used to what it's okay to have in solution. So tell me what you're doing in the Center for Misfolding Diseases. I see someone working behind you, so I'm impressed.
Yes,
They're still there.
I think it's all right. Yeah, so these days we have a lot of lines of research. I'm a member of Michele Vendriscola's lab.
Okay.
And I'm a part of a team that is working on designed antibodies that are rationally designed to bind misfolded or unfolded proteins.
Very nice. Okay, thank you.
Thank you very much.
Pleasure.
Oh, there's another question in the chat by Yiha.
Hi, Yiha. Are you still there? Hi, Yiha, anyway. So, yeah, you ask about whether or not we could have an automated algorithm and that if the structures are so diverse. And yes, I think is the answer. So I spoke with Shaw Shares, I'm sure you probably know, at the LMB, and he keeps telling me I should be using AI to do some of these assignments because they're so complicated. But what would have to happen would be that somebody would have to, they probably wouldn't like to, but they would have to sit with me for a week and kind of learn how you go about doing something that's a bit routine but has rules around it. And sometimes I look at it and I know that a particular assignment isn't true, but I can't always say why I know it's not true. But there must be a rule, there must be a set of rules. So it's about trying to teach an AI algorithm those rules, I guess. I just need to spend a week or so sitting with someone who's skilled at that, and I'm sure it could be possible. And you also ask about whether it would be great if you could describe how your company span out, how it operates, who are the clients and what they do. Yes, I think it is interesting actually to do this. I mean, I've never before fancied having anything that wasn't just academic because I felt I couldn't really cope with anything else. But more recently, I realized that actually I don't have to run it. I get someone in who knows much better how to run a company than I do. So I guess it was the GPCR work. We started to get a lot of interest from companies, and that started off as collaborative projects. And there was always a lot of IP, and in Oxford it's always quite difficult to come to these IP agreements, and it would take ages to set them up. And somebody said to me, well, if you just put your IP in a spin-out company, you negotiate the contracts and you go from there, and you don't have to keep going through the university. You just got to buy your IP and put it in your company. So, well, that's actually sounds like a much tidier way, because the agreements would take so long that by the time we'd got them in place, the company sort of said, oh, well, we're not doing that anymore, we're not interested. So it was really for speed, I think, that we did it in the first place. And then we were going to raise some funding to expand. It was going quite well. We're making quite a reasonable profit with this kind of consultant company, I would call it. And then Syncona, who were part of the Wellcome Trust, said, well, actually, you should be more ambitious. You should have your own programs that you do in-house, find your own molecules and then carry those through. So that's what we now do. We very fortunate we had a great CEO from Boston, Ros Deegan, and she had run GPCR companies before, so she knows what she is doing, and we've all learned a lot from her. And I go in as a consultant, but I don't run the company. But yeah, I think it is an interesting kind of relationship, and it certainly changed the way I think about research, because you suddenly see how useful it could be in diseases, which I hadn't really thought about so much before. So yeah, I'm enjoying that particular journey.
Right. Thank you so much. Are there any further questions from the audience? Maybe if it's okay, I can ask a second question.
Sure.
Yeah. So maybe just to follow up from the previous question, and also during your presentation, you mentioned about, I think there were, you mentioned something like there were 3 different papers by Science, Nature, and Cell, which showed different stoichiometries due to the way that the crystals obtain or the detergents they use. So how reliable do you think are the old structures determined by X-ray crystallography, particularly for membrane proteins?
Well, I always think that, you know, you do your best with the tools and techniques that are available at the time, and we're always learning. I think none of them are wrong. They're just different views of a particular complex. And so, in fact, 2 of those were done by the same lab. So they first produced the monomer, and then another group, the dimer, and then the original lab went back with the tetramer. So I think it's perfectly fine to reevaluate things in the light of new information, and I don't consider things being right or wrong. I just think it's a different view, different conditions, gives you a different view of a complex, and that's all fine in my opinion.
I guess that's the advantage of native mass spec, where you can get it as close as possible to how it's like in an actual cell.
Yeah, although some people would argue that it's not, you know, so there's always a question about every technique, I guess, that people use. So, yeah, of course, we believe in what we're doing, but, you know, other people say, oh, no, it must be this, or you know, it's always open to interpretation. But in my opinion, all of those views are very valuable.
Thank you very much.
Okay.
Are there any further questions from the audience?
I think people are off for their dinner.
Yeah,
I don't blame them.
I think so. Then maybe I'll just wrap this up then. So if there are no further questions from the audience, we'll now bring this lecture to a close. And thank you so much, Professor Robinson, for giving us this really exciting and inspiring talk about your research on mass spectrometry. I really enjoyed this talk. And last but not least, thank you to members of the audience for joining us today, and as well as for being a regular supporter for our online talks this year. Unfortunately, we didn't have any in-person talks. But thank you so much for supporting us despite COVID.
Yeah, thank you very much for being a great audience. And yeah, I would have preferred to come, but this is the best we can do. So yeah, good to see you all, and good luck with all your research. And I guess you're all off on holiday, well, break soon. So yeah, have a good break. And thank you again for the invitation.